All right. Hi, everyone. It's my pleasure to introduce our speaker for today, who is Andrew Camara. Andrew is a PhD student at the University of Warwick, where he's completing a joint PhD within the University of Warwick, Monash University joint PhD program. He, in his PhD, is working on a bunch of stuff to do with judgment and decision making and sports betting. He kindly agreed not to talk about that today because we have another talk from someone else in our lab later in the semester on a similar topic, and we didn't want to clutter up your seminar series with our work. But Andrew's kindly agreed to talk about some stuff that he did in a previous RA position. So take it away. Oh, and I meant to announce that there will be, in lieu of a lunch with Andrew afterwards, there will be a social CHDH social pizza lunch on the 8th.
Be around here for several months.
That's right.
Yeah.
So Andrew will be have a desk here along with a couple of other folks from our lab who are moving down. So yeah, this is what he looks like. He's friendly. Say hello to him.
Okay, thanks for having me here. Yeah, so today's talk, it's going to be on incentives, response inhibition as measured by the stop signal task, and intracortical measures as assessed using transcranial magnetic stimulation. Yeah, it's a research, it's from a research assistant role I had at the University of Sydney. Basically, I did the experiments, left the position, but I told Dom, I was like, hey, I'll finish off this analysis and submit this to a journal soon, hopefully. That was in 2023. And so I thought I would use today's talk as a way to stage an intervention against myself, where we'll use the fear of social embarrassment as a way to motivate me to work on this project. I can confirm its work, very scared, but we have some shiny new plots that I can show you. And so the kind of background motivation for being involved in this area is to realize that when we use response inhibition in the real world, it's often in connection with a kind of highly salient stimulus, which is encouraging us to approach it. So whether it's in gambling or saying not to eat junk food, when we are engaging in response inhibition, which is just basically inhibiting a response tendency that you have because it's contextually inappropriate. And we're doing it because there's something there which is motivating us to make a response, okay? And this is something which doesn't occur when there is not a kind of highly incentivizing stimulus there. Like if I was to dangle a vegetable in front of your face, you're not going to kind of actively engage your inhibitory processes to stop yourself. And so the idea was to try to investigate this in an experimental context. The task we'll be using is the stop signal task. If you haven't heard of the task, it's basically got two main types of trials. On most trials, there is a go trial in which you just you perform a speeded response to a fairly simple stimulus. In our task, all participants have to do is when they see a right arrow pop up on the screen as quick as they can, they press the right arrow key on their keyboard. When a left arrow pops up, they press the left arrow key, and so on and so forth. Now, that's what happens on most trials, but on a minority of trials, we have stop trials, where after the go stimulus pops up, a stop signal then appears, and you have to ignore what the go stimulus says and do nothing. In our experiment, what this looks like is after the arrow pops up, a square then pops up on that arrow. If you see the square, then you ignore what the arrow says and you don't do anything. So it looks something like this. And so the idea is that you see the arrow pop up, you start to make a response, but then when the stop signal appears, you have to hold yourself back. And so it's a measure of how quickly you can cancel a response that you're about to make. So previous research which has looked at the role of incentives in the stop signal task have mainly manipulated it in two main ways. Number one is that you can inform participants before the trial starts whether or not an incentive is available. The results vary a little bit depending on how exactly you implement the incentive. So if you incentivize people, for example, to correctly perform a stop trial versus a go trial or vice versa. But generally speaking, you can increase a person's stopping ability by offering an incentive like this. Another way you can do it is that you can do it by using features of the stop signal to signal whether or not an incentive is available on that trial. And again, we find that people are generally better at stopping when the stop signal indicates that an incentive is available. Now, what we want to do is we want to try to do a similar thing, but using features of the go signal. So in our experiment, the color of the arrow which pops up will tell participants whether or not an incentive is available. And this is kind of interesting for two main reasons. Number one, I think it kind of tracks better with what we were talking about earlier, where the thing which we are trying to inhibit a response towards is the thing itself which is motivating us to make the response. The other thing is that it's not necessarily clear to me that signaling incentives at this stage will make people stop better. The kind of reasoning being is that if you're using something like the stop signal, there's only kind of one thing the person can do at that point, which is to stop. But at the go stage, the person will either go, but then they might also have to stop as well. And there's some evidence that when you use features of the task itself to signal whether or not an incentive is available, generally people will engage in an approach behavior when there is an incentive, and they'll be less likely to engage in a withdrawal behavior. And so we thought that people might perform better on go trials, but then actually perform worse on stop trials. And so that kind of leads us to our first hypothesis, that signaling reward availability via the go queue will impair stopping performance. Our next hypothesis has to do with our intracortical measures. So it's using something called transcranial magnetic stimulation. So basically, you just take a big coil, put it over a person's head, and you send an electromagnetic pulse that will then activate the neurons over that area. And in our experiment, what we'll do is that we will stimulate a person's motor cortex, and we will measure the response from their hand. The response that we get is it indicates the level of excitability in their motor cortex at that moment in time. And particularly what we're going to be doing is we'll be measuring their level of inhibition using something called short intracortical inhibition. So in your kind of standard pulse, what we'll do is we'll give a fairly strong pulse. So that's 120% of their resting motor threshold, which is just it's the minimum stimulus intensity that you need to use to have a 50% chance of getting a detectable response. So we go a bit higher than that. And what we'll do is, before we give that single pulse, we'll actually precede that by a smaller pulse, which isn't strong enough to activate the output neurons themselves. What we then find is that the response from the person's hand is actually smaller than when you just give them one pulse. And the thinking behind this is that this weaker pulse, it's not strong enough to turn on the output neurons, but it is strong enough to turn on these GABAergic neurons whose job it is to silence those output neurons. And an important thing here is that people vary in the degree to which this inhibition is present. So it'll be fairly strong in some people and it'll be weaker in other people. And what previous research from our lab has shown is that this variation in Psyche will predict how well someone will perform on the stop signal task. So I've plotted, so Psyche here is plotted on the y-axis. Quite counterintuitively, the higher someone's Psyche value is, the worse they are at inhibiting their motor cortex. And I've got stop signal reaction time on the y-axis. So I'll explain that a little bit more later, but essentially higher values here means that the person is slower to stop on a stop signal trial. And you can see that the worse that someone is at inhibiting themselves, the slower they are on stop signal trials. Now, this is Psyche measured when a person is at rest, when they're not doing anything. Another thing you can do is that you can measure a person's Psyche over the course of the stop signal task. So here again, Psyche is on the y-axis, and the time since the stop signal has being presented on a stop signal trial is on the x-axis. The thing I want to draw your attention to is the red lines here. So the solid red line are fast stoppers, so people who are good at stopping on a stop signal trial, and the dashed line are people who are slower. And what you'll see is that if you're fast at stopping, initially there is a weakening of Psyche that's basically in response to the ghost stimulus, but then the person is quickly able to bring Psyche back into line and kind of turn on that Psyche brake. Whereas people who are slow, they experience that same loss in inhibition, and they're never really able to get that baseline level of inhibition back. And so it seems that people who are kind of faster on this task kind of have this extra inhibitory tool in their belt that they can use. And so with this line of reasoning is that we've thought that if someone had a strong level of intracortical inhibition, that they might be somewhat protected from any bad effects if there is an impairment when we use incentives in our task. And so there's our two hypotheses: signaling reward availability via the go queue will impair stopping performance, and those with stronger Psyche will be less vulnerable to this effect. So that brings us to our first experiment. We got 27 undergraduates, a fairly small sample, and we measured their baseline Psyche, and then we gave them a rewarded stop signal task, where the color of the arrow let them know how many points were available on that task. And they were either a low value trial in which there were 5 points, a high value trial in which there were 20 points. And we also had these jackpot style trials, which weren't that frequent during the task, but which had a high points value associated with it. Just trying to see if there was any kind of extra effect of kind of really boosting up the salience. Importantly, people got these points just for doing the task correctly. So if it was a go trial and they made the right go response, they got the points. If they stopped on a stop trial, they also got points. So we're not trying to bias people towards prioritizing one task type over another. Now, before we get to the findings from this first experiment, we should talk a little bit about how we measure people's stopping ability in the stop signal task. The main measure is stop signal reaction time. Now, this is just how quickly a person can respond to the stop signal and stop themselves. It's somewhat hard to measure because if a person stops on a stop signal trial, you don't know when that inhibition process finished. But what we can do is kind of exploit the facts. We assume kind of a fairly simple model of how a person stops, or in this case, fails to stop. So if they commit a commission error, which is just when you make a response on a stop signal trial, it's assumed to be a race between a go process, which can be measured by your reaction time on go trials, and this stop process, which in order to figure out when that finishes, you need to know the stop signal delay. This is just the time between when the go stimulus appeared and the stop stimulus appeared, because of course you can't stop until you see the stop signal. And then how quickly you respond to that stop signal, your stop signal reaction time. And what we do is we exploit the fact that we can't measure this, but we can measure everything else, and we vary the stop signal delay across trials based on how good a person is at stopping. So if we have a stop signal delay here of 150 milliseconds, and let's say I fail to stop, on the next trial, that'll be made shorter. So the stop signal will pop up earlier. So then I'll get kind of earlier warning that I need to stop. And let's say if I'm successful on that, then it'll be bumped up again. And by varying that over the course of the experiment, you're able to kind of converge on a stop signal reaction time. And when you're using a procedure like this, on average, people will have a 50% commission error rate. Now, we're not using that in the first experiment, and that's because we used a joint staircase, which is just where how we changed the stop signal delay was blind to the points value associated with that go queue. So if a blue arrow popped up and I failed to stop on that, the next stop trial was made easier, independent of what type of trial it was. So if it was a blue arrow, it was at 100 milliseconds, but equally, if it was a yellow arrow or a red arrow, it was still the same. So what happens then is our commission error rate now becomes our primary dependent variable as opposed to stop signal reaction time. To kind of flesh this out with an example, say we have a fictitious participant here, and they conform perfectly to our hypotheses, and they are fastest on a low value trial. They're a little bit slower on a high value trial, and they're really slow on a jackpot trial. Assuming that we have the same stop signal delay, they should have a low commission error rate on low value trials, a little bit higher on high value trials, and they'll be worse on these jackpot style trials. Now, unfortunately, I was unable to use a fictitious sample of Daniel Bennett's to run my experiment. I had to find actual participants, and that proves to be quite a key mistake in this experiment. So here I've plotted commission error rate on the y-axis, and on the x-axis I've just got the different incentive conditions. And the only thing of note here is that there's a bit more variability in the jackpot trials, but that's kind of to be expected because there's just less of those trials available. But overall, there's not really a consistent pattern emerging from this. Although our task isn't kind of it's not designed correctly to measure stop signal reaction time, it is worth noting that we also don't get any effect if we try to measure that. Turning to the other kind of behavioral measures that you can pull out from the stop signal task, one is the emission error rate. Now, this is just where the go signal pops up, but a person fails to make a response to that. And so that's shown over here, and we did find that jackpot style trials were associated with a lower emission error rate. So we had to use a parametric test because we get quite a strong floor effect here. But what we found was that on jackpot style trials, that people had a lower emission error rate compared to low value trials, with high value falling somewhere in between, but not really turning out. Turning to our TMS measures, so SICI on the x-axis, I've plotted SICI here, and just as a reminder, as this value increases, a person is worse at inhibiting their motor cortex. And on the Y value, we have a commission error rate. And we'll see that in the low value trials, there's a tendency towards a person who has weaker SICI having a higher commission error rate, which kind of makes sense. On high value trials, this seems to flip. So a person who has worse inhibition will actually have a lower commission error rate on these trials. And on jackpot style trials, it's fairly flat. What we did to kind of properly test this was we ran a linear regression where we use Commons contrasted coefficients. So we had one comparing the commission error rate on high and jackpot trials versus low value trials, and then one comparing jackpot and high value trials. We found only the first one was significant, such that if someone had weaker inhibition, they made less errors on high and jackpot style trials, which isn't exactly what we were expecting. One thing to note here is that this overall model isn't significant, so it's not a particularly strong finding. It's kind of just trending that way. Yes.
I've got a really dumb clarifying question. Commission errors here, thinking through where I should have stopped, but I didn't.
Yes.
So I didn't respond to the stop signal.
Yeah. Yeah, so it's not an overly strong pattern of findings. So to kind of summarize, we didn't find much evidence for an effect of incentives on stopping performance overall. We did find that it does seem to facilitate the go process to an extent. And we see some evidence that SICI might moderate the effect of incentives, although in a fairly unexpected direction. For our second experiment, number one, we got a larger sample, so we had 49 undergraduate students. We then tried to simplify the stop signal task. So we got rid of the jackpot style trials and we only had the low and high value trials. This was primarily because we wanted to measure stop signal reaction time, and that's just hard to do if you don't have a lot of stop trials. And we used separate staircases for low and high value trials, so then we were able to better estimate stop signal reaction time. So if someone failed on a low value trial, only the next low value trial was made easier. It didn't affect the high value trials, and the stop signal delay on that was set separately based on your performance. What we did as well is that in addition to measuring baseline SICI, was that we also measured something called SICF. So just to recap, so we've got our standard single pulse here, which generates a response in the person's hand. If we proceed that with a smaller pulse, it turns on these inhibitory neurons such that there is a weaker response. What happens in SICF, which is short intracortical facilitation, is that we mainly just swap these around. We use slightly different parameters. So we give the big pulse first and the small pulse second. And what we find is that instead of a weaker response from the hand, it's actually stronger. Now, the thinking on this is that when the first pulse comes around, is that it turns on some but not all of the output neurons. And in particular, there'll be some neurons which come close to firing, but which don't quite get there. And so when this weaker pulse comes along, it turns on those neurons and it contributes to the response. Now, the reason why we decided to measure this was based on a previous study which looked at stop signal reaction time and SICI, and they didn't get a great correlation. They measured SICF and stop signal reaction time, and they got a better correlation. And so we added this measure in to see if kind of this was a better measure in our task. Turning to our findings, so these are the behavioral findings. So stop signal reaction time is now plotted on the y-axis across the low value and the high value conditions. Again, we don't really find an overall effect. We also ran a Bayesian analysis as well, and the again, turning to the other measures that you can pull from the stop signal reaction time, like in experiment 1, we find an effect on emission error rate. It's in the direction that we would expect. So people are less likely to make an emission error on a high value trial, which is to say that in a high value trial, you are more likely to respond to the go signal. Before we turn to our TMS findings, one thing to note is that there is a correlation between SICI and SICF. So where the weaker someone's inhibition is as measured by SICI, the stronger their facilitation will be as measured by SICF. And this is kind of largely thought to be due to the fact that they're measured in quite a similar way, right? Because you're giving two pulses close together, one's a bigger one, one's a smaller one. And so they're kind of not pure measures of each other. So when you use SICI parameters, you are going to get a little bit of facilitation as well. When you use SICF parameters, you're going to get a little bit of inhibition as well, such that some people, they suggest that when you're looking at these measures that you should kind of examine them together so then you can tease out the individual effects. And so what we're going to be doing is we're going to be throwing both of them in a linear regression where they will try to predict the difference in stop signal reaction time between low and high value trials. So those plots are, so the plots of the correlations are shown here. On the x-axis, I've got the level of inhibition / excitation, such that the more that we move to the right, we get weaker inhibition and stronger excitation. And moving to the left, it's stronger inhibition, less excitation. And on the y-axis, we have stop signal reaction time. The way that the panel's split up is that on the left side, we've got our low value trials. On the right side, we've got our high value trials. The top row is SICF and the bottom row is SICI. If we look at the top two panels first for SICF, you'll see that's There's not really much change in the slope of the line between the two plots, and in both cases, they're not significant. If you look at Siki as well, on the bottom row, you'll see that again, both the correlations aren't significant, but you do kind of get this change in the slope between the two conditions. And so looking at the coefficients in our linear regression, we see that sec-f isn't a significant predictor, but Siki is. Again, it's in a somewhat counterintuitive direction where the stronger someone's Siki is, the worse they will perform on a high value trial. And again, we unfortunately find that the overall significance of this, significance of the model, it falls slightly short of significance, which is to say it is not significant. Another thing to note as well is that I say it's coming out in an unexpected direction. It's not quite the opposite direction to what we were expecting. One thing is that I'll just kind of plot kind of what this does look like. So if I plot on the y-axis the difference in high-value trials and low-value trials, and on the x-axis in Siki, you'll see that most people are falling into the quadrant on the top left or on the bottom right, where in the top left, if they have strong Siki, if they're good at inhibiting their motor cortex, paradoxically, they seem to perform worse on high-value trials. When they have weak Siki, when they're not good at inhibiting their motor cortex, they're faster on these high-value trials. And it's worth just going back to what we were expecting in our experiment. So with our fictitious participant here. So what we were expecting would be something like a mean difference in stop signal reaction time across our Siki groups. So where people with a high level of Siki do better on the task on average, and it's fairly consistent across the low-value trials and the high-value trials. Whereas those who have low Siki, they're worse on the task overall, and they're particularly bad in the high-value trials. What we get instead is something that looks a little bit more like a crossover interaction, where on average, there doesn't seem to be an effect of Siki, but then underneath this, for our low Siki group, they seem to do better as you go from low to high incentives, whereas our high Siki group, who are good at inhibiting themselves, when you turn up the incentives, they seem to do worse. Okay, so just to summarize, overall, we didn't find much evidence for an effect of incentives on stopping performance. There's some evidence that Siki moderates this overall effect in an unanticipated direction. I haven't got it here as well, but it is also worth noting that we do at least find an effect of incentives on the Go process itself. Okay, I'm going okay for time, so I might kind of talk a little bit about where I want to kind of take this analysis in future. So in recent years, there's been a reconsideration of how people analyze the stop process, where if someone fails on a stop signal trial, so if the stop signal pops up and I fail to stop, it could be, broadly speaking, due to at least two things. Number one is that my stop process could just be slow. It started, but it didn't get there in time and made the response. Another thing is that it just could be the case that I actually failed to trigger that stop process in the first place. And this is in part motivated by experiments where even if the stop signal pops up quite fairly early, so if the go signal and the stop signal pop up at about the same time, at least on some trials, people will still fail to respond to the stop signal and make a response, which kind of indicates that at least on some of the trials, people are just not initiating this process. So we're talking about kind of the consistency of being able to engage your stop process versus the overall speed. using Bayesian modeling, you are able to kind of tease out these factors. And what research has shown is that previous effects, which we kind of pinned on stop signal reaction time, so a slower stop process, was actually due to differences in the trigger failure rate. A notable example being the effect of signaling incentives via the stop signal. Turns out that effect is not completely, but largely due to actually less trigger failures. So what we have done for the experiment so far is that I've got overall measures of these parameters just for our behavioral findings, but I haven't associated these with our TMS measures yet, which is just because in Bayesian modeling, rather than getting a point estimate, you get a distribution of values, so it's a bit harder to analyze. But when we look at SSRTs after accounting for trigger failures that might be present in the data set, for experiment 1, we still don't get a difference in stop signal reaction time. What we do find is that people are more likely to make a trigger failure on a jackpot trial. Now, I'm a bit tentative about this finding just because, as I mentioned at the start, we don't have a lot of jackpot trials in our data set, which could have affected the estimation process. It's also worth noting that the effect might be in part due to a kind of subsample of participants who have a really high trigger failure rate. So just for context, the average trigger failure rate in non-clinical samples is around 4 to 18%. Although you can get quite high values, especially in clinical samples. But yeah, nonetheless, that's the general pattern of findings that we get. Fortunately, when we turn to experiment 2, when we're just looking at low and high value trials, looking at stop signal reaction time after accounting for trigger failures, we still don't get a significant difference. And that's the same with trigger failures as well, although it does seem that there's less of a range in the rate of trigger failures in the high value condition, although that's just kind of just from eyeballing these point estimates. So, turning to a discussion of our key findings, kind of the most consistent thing which pops up across the 2 experiments is that when you provide an incentive, you get a facilitation in the go process such that people are more likely to respond to the go signal. Now, unfortunately, there's not too much to perhaps, or at least there's less research available to aid interpretation here because go emissions aren't really as extensively explored as a stop process in the stop signal task. Largely, go emissions are more seen as a nuisance variable that you need to control for when you're trying to investigate people's stop process. Nonetheless, it's generally thought to be some kind of an attentional failure, although kind of where this occurs in the process isn't exactly well known. So it could be more at the early stage where people just kind of fail to encode the go stimulus, or it might be something like a trigger failure, but for the go process, where people just actually fail to initiate a go response when presented with the cue. So there's at least kind of 2 possibilities there. Turning to our stopping findings, we found consistent evidence that overall stopping performance wasn't substantially affected by incentives, although there's maybe a higher rate of trigger failures on these kinds of jackpot value trials. And we found some evidence that this overall kind of null finding might mask a bit more of a complicated relationship with Siki. Now it's a bit hard to interpret, and I'm not too sure exactly what to make of it. The kind of best I can offer at this stage is to kind of draw your attention back to this experiment here, where they measured people's level of inhibition over the course of the stop trial. And we found that there were some people who were kind of faster on the task and they were kind of able to engage their Siki break, whereas there were people who were slower and they kind of weren't able to use that tool. And the point to kind of make about these people who are slower is that they still do stop on some trials. It's not as though Siki is the only thing at play here. And so perhaps the reason why we're getting a bit more of a nuanced interaction here is that there might be kind of other inhibitory pathways involved apart from Siki, which add some nuances to how people are affected by incentives. Beyond that, since I don't really have a sense of what those might be, it's kind of the best that I can offer up at this point. Now, where I plan to take this research in future, I would like to look at the relationship between these intracortical measures and trigger failures, just to see if there's something there in terms of failing to initiate the stopping process. And then ultimately, I would like to explore previous research a bit more, just to see if there's kind of other parallel examples, maybe not necessarily in intracortical measures, but just other measures of inhibition as well, where we find this kind of counter intuitive effect. Like I said at the start, this is an intervention that I've organized against myself, and a key part of any intervention is when the person involved realizes that they need help. And so if anyone here, I suppose, has familiar with similar effects which might kind of track well with what we're seeing here, I would be excited to hear about it. And yeah, that is the end of the presentation. Yes,
Just have a-
sure.
So I'm new to this topic, so I don't know. So is there any kind of like previous work that looked at use this similar tasks, but then they have this like training period where people like feed on this like stuff in this task so that they can get better. So I'm just thinking, because in real life, we think junk food, of course, there's a reward to eat, healthy food, but it just takes this habit, like this conscious habit to make. So I'm just wondering whether this training versus reward.
Oh, yeah. So, I mean, you'll at least in the task, like you will give them like initial training phase, just to give them familiar with the task. I know there's, they have tried to kind of train people on the stop signal task. From memory, I think they get at least some success with doing that. Within the context of incentives and how that tracks, if that influences things, I'm not too sure. I know people have tried to use this in things like substance use, for example, where they'll take a clinical sample and they'll kind of train them on substance kind of stimuli, and they will practice a response to that. What the overall effect is there, I can't remember, sorry. I'm not too sure. There are at least some studies showing success. Whether overall in a meta-analysis, for example, if you would still find an effect, I'm not too sure. No worries. Yes.
Good talk.
Thank you.
I had sadly experienced with stop signal. I collected a lot of the data for the skin paper.
Okay.
So first of all, I feel for you. Stop signal is a notoriously difficult thing to get at.
Yeah.
One question I had about what you show that has the Siki response over time.
Yes.
Was that collected using a single reward structure? Like there wasn't kind of different conditional changes between them?
I don't think so. I think that was just a standard version of the task.
So my question is then, do you think that some of the issues you're seeing is that people are responding primarily to the high reward structure, and as a result, the low is kind of copying it. Like it's not really getting the response we'd expect out there. So another way of doing it, if you was to say block those two sets,
Right, okay.
would you expect maybe to get a cleaner, better result that aligns with what your kind of expectations? Because trial to trial, they're not being sensitive to that reward structure in the way you would expect. This is my thing is, as you show your stuff, like concept thought, it seems to match up with what you show.
Sorry, so just to to try to summarize that, so because you're jumping between these high value and low value trials, people are prioritizing the high value trials, and so then as a consequence, it's worse on the low value trials. And that might be a reason why someone who's otherwise good at the stop signal task performs worse on it.
Yeah, and it would still keep a nice, like high and low kind of diagonal where the individual responses to it.
Yeah.
But the interaction terms of the high and low themselves would kind of get muddy.
Yeah, okay.
I suggest this because we're doing work in visual working memory stuff, and we're seeing a lot of weird global experiment effects where if you start pulling things apart, you get the effects that you expect, and when you start mixing them together, they start.
Yeah, okay. Yeah. So I guess I'm also just thinking of, how that works for the inverse of, so I can see why, like, if someone is good at stopping, why giving an incentive there might mess things up. I guess I'm wondering just how that works for the flip side, where if someone's poor at stopping, why then you do at least trending towards getting a benefit for an incentive. I mean, I guess maybe it's because they're less able to kind of modulate their stopping process, so then you might get findings which are in line with it. I think another tricky thing as well is that if you are going to block it, I suppose the value of signaling whether an incentive is available at the go signal kind of loses meaning then, unfortunately. But yeah, I think that does track, and that's an interesting explanation, yeah. But yeah, not too sure how I'd pull that apart in my data set per se.
And but
Yeah.
something to look at then is if the previous work is showing this kind of stuff, if their structures are not kind of a mixed design and they get the results that you would expect, maybe that could be an insight into why it doesn't happen.
Yeah, definitely. Yeah. I should go back into the literature looking at blocked versus intermixed trials. Cool, thank you. Yes.
My question is like obviously involved in performance. So you have this idea that TMS is targeting GABAergic neurons, is that correct?
Yes.
Yeah. What do you think GABAergic neurons have to do with reward? Like, how do you think they're influenced by reward relative to dopaminergic neurons or noradrenergic neurons, for example? I'm just trying to understand, like, I guess, like, naive stuff, why your hypothesis is with those specific ones, because I can imagine reward facilitates dopaminergic and noradrenergic stuff, but I just didn't get think out like that. So.
Yeah. I mean, suppose it was perhaps maybe not directly involved, but I guess just more from the fact that if there is some kind of detrimental effect there, a kind of person who also has this inhibition present kind of might be more protected from it. I think another thing to note here as well is that so you do kind of get studies like this which kind of seem to provide kind of a mechanistic explanation, but you are also just kind of measuring, I guess, a fairly kind of late stage of the inhibition process. So it could be something kind of further along in the chain, which is kind of driving this effect. Beyond that, Yeah, sorry. Yeah, I don't think I can really comment on that further.
When you see them in motor cortex
Mm.
and they're projecting to like higher order frontal regions and that's doing the inhibition, or is it like I'm doing it to the motor cortex and it's just like I'm about to press the button and that's what's happening? So do you know what I mean? Like at which point the TNS is doing it?
Yeah.
Is that a weird question?
Uh, no, it's not. Yeah, sorry, I'm just not sure I have a good answer to it. Sorry.
Question.
Sure.
She said, nice studies and presentation, thank you. I was wondering whether the reduction in emission errors with rewards, so improvement in go performance, could be considered optimal. If you think about the fact that there are 75% go trials, so if you may get a reward, maybe you are best off trying to improve your go performance rather than stopping performance on less frequent stop trials.
Yeah, so that is something I considered as well because there are some yeah, where there are some previous experiments where they will make a stop trial worth 3 times more than a go trial. I think so. I mean, I guess it's a bit hard to unpick from a participant perspective as well, because participants generally fail more with the stop trials. So a kind of big frustration in the stop signal trial is, I guess, trying to get participants to actually go fast on these go trials as well. Um, yeah, because they'll kind of tend to prioritize the stop trials already, and so I'd be a bit worried about kind of changing the reward structure based on that. Um, but yeah, I think I do ultimately concede that it is kind of a good point to notice that if you did just try to prioritize go trials, um, yeah, then that would kind of be a better strategy, particularly as well because how the stop to signal trial works is that it tries to make it so that you fail on stop trials 50% of the time, no matter what. So you can't really uh, improve the stop trial via your own strategy. Um, yeah, I hope that answers the question. Yeah, I'm not sure to, yeah, no worries.
I
Yes.
have a related question, or kind of related topic, which is the change in rates of emission errors is kind of an interesting, consistent finding. There's another, you focus mostly on errors, but you could also theoretically measure the rate or the vigor of the successful go responses. Do you find, for instance, that when rewarded the higher, people faster to go on the successful side?
So in experiment 2, no. In experiment 1, it trended towards people being faster on jackpot trials relative to low value, but then from low to high. Wait, sorry, I need to actually look at that. I think they were slower. They were slowest on the jackpot style trials. And they were a little bit faster on the high value trials. So yeah, so on the low value trials, it's around 416. It dips to 411 on high value trials, and then it's slower on jackpot style trials.
It's actually quite striking then that the emission error rate goes down by a factor of more than 50%, but without any corresponding cost in that. Because you might expect one reason why there's the emission error rate changes is that the overall distribution gets shifted. What you're saying sort of implies that the distribution isn't getting shifted with the error rates going down, which might be a more efficient processing.
Yeah. And again, not that we can exactly do the analysis to pick this apart, although there is a kind of Bayesian modeling that you could do. But yeah, it could be something, yeah, in terms of like a lower trigger failure equivalent for the Go trials. We did try to explicitly account for this in experiment 2, just because we had kind of emission error rate distribution, which didn't have as much of a ceiling effect, where we ran a linear regression where we controlled for the difference in response speed across the two conditions. And yeah, you find that that does predict the rate of emission errors, but even when there's no difference in response speed across the two conditions, you do still find this residual effect on emission error rate. So yeah, there does seem to be this influence over and beyond kind of the speed of the Go process itself. Yes.
My first question, it's just about the question of Paul's idea about like working versus like randomized trial. I'm kind of struggling to understand why do you feel that by working the high low reward rate would kind of lose the meaning of high reward. Like if the like for the participant, the goal is to maximize the point over the whole session, then it should not make much of a difference. And associated reason for perhaps considering about working over random trial design is the temporal delay effect from one trial to another. And like, I guess, like was a generally speaking, like the temporal effect such that like, for example, success or a failure in previous trial has an effect in the subsequent trials is kind of everywhere and everywhere, right? So we can like just disregard it or not. But I feel like a specific reason why we might concern about this in this scenario is that I would intuitively think that fail in a high reward trial has a different effect at the first trials compared with a fail for a like low reward.
Yeah.
Trial. And just because that high reward trial is you are kind of like easier to fail, it means that if say you fail high reward, follow a low reward, you are kind of manually boosting up your performance for low reward trial just because you lost a big fish just now. Overall, you know, kind of if which I guess I'm saying like with random trial design, you are like unconsciously boosting out your low performance, like low reward performance.
Yeah. Yeah. So I think my main concern with putting it into blocks is so that I guess just in comparison with previous research where they kind of signal the availability of an incentive before the trial starts, I guess the idea was that we've kind of mapped that space a little bit, and I wanted to see what the effect was when people find out whether or not an incentive is available at that go stage. I think it would still be interesting to block it just to see if there is an effect of kind of that stimulus difference there, even if participants don't need to attend to that. But I think that's a good point about the carryover effect from the different trials. I was tempted to pull out something, like post-error slowing. Which for those of you who kind of haven't heard of that before, that's just when you fail on a stop trial. And so because you're kind of more cautious of making a response on the next stop trial, you then try to slow yourself down. And I hadn't considered that point before. I was kind of a bit hesitant to try to go looking at things like that because I was worried that this was just going to kind of transform into a phishing exercise. But I think that's actually a good point as to why we should try to look at that. I'll look into it. Yes.
Let's make this the last question.
Sure.
So we can continue the discussion over pizza.
Yes.
I think that would be good.
Just looking at your plot there.
Yes.
Because the RT thing is weird, but RTs are stable, error rates going down. One possible explanation, and maybe Laura wrote about this in her paper, would be a joint collapsing down model that's sensitive to urgency and improved drift rate with your jackpot signal. So that would allow the RTs to be stable while the error rate would improve. I can talk to you about this after, but
yes,
it would be, I think there's a way of explaining why you might see this in fails.
Yeah. Oh, cool. Okay. Yes, I'd be keen to hear about that. Unfortunately, so the so the Bayesian modeling stuff that we've been doing so far, that's been a co-author of mine. So I'm kind of yet to get kind of stuck into that properly myself, hence why I was a bit hesitant on that stuff. But yeah, I'd definitely be keen to explore that.
In any case, thank you very much. And
No worries, thank you.
yeah.